OCC-1. OCC-1 overexpressed in Carcinomas 1 is a protein which in humans is encoded by the gene C12ur75. The gene is approximately 40,882 BP long and encodes 63 amino acids. OCC-1 is ubiquitously expressed throughout the human body. OCC-1 has shown to be overexpressed in various colon carcinomas. Novel splice variant of this gene was also detected in various human cancer types, in addition to encoding a novel smaller protein 51 amino acids. OCC-1 gene produces a non-protein coding RNA splice variant lncRNA called OCC-D variant. Gene location and size. C12ur75 is found along the plus strand of chromosome 1212 at 23.3. The gene is 40,882 BP long with the genomic sequence beginning at 105,330,636 BP and ends at 105,371,518 BP. C12ur75 contains 6 exons and is flanked by KCC8-198 renal clear cell carcinoma-associated transcript 198. mRNA, RNA, 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 RNA. C12ur75 encodes several transcripts of mRNA that the longest one is 1386 BP. The mature mRNA of this splice variant contains 6 exons. Protein structure. Primary structure. OCC-1 protein is 63 amino acids long and has a molecular weight of 6.4 kDa. OCC-1 contains an opioid growth factor receptor repeat OGFR motif from residue 8 to 27. OCC-1 is an acidic protein with an isoelectric point of OCC-1 is 6.6. Secondary structure. The secondary structure of OCC-1 is a combination of multiple coils, a few helices, and few B sheets. The Fire2 program 52% helices, 6% B sheets, and 70% disordered. The predicted region B sheet from residues 31 to 38 coincide with the helices and B sheets regions predicted by other programs. OCC-1 is a soluble protein according to the SAUCI program, therefore there are no transmembrane domains. Tertiary structure. Predicted folding by ITASR is shown. Post-translational modifications. OCC-1 is predicted to undergo the post-translational modifications of O-glycosylation, phosphorylation, and myristoylation. Subcellular location. The KNN tool places OCC-1 in the nucleus of the cell with 65.2% certainty, 13% in the mitochondria, 8.7% in the vesicles of the secretory system, 4.3% cytoplasm, and 4.3% vacuole. Homology. Paralogs. OCC-1 has no known paralogs. Orthologs. OCC-1 has been found in Mammalia, Reptilia, Amphibian, Abs, and Actinopterygiae. The gene is not found in plants, protists, fungi, archaea, or bacteria. The most distant ortholog is the Oryzias latipers, or the Japanese rice fish, which diverged from the human gene approximately 436.5 million years ago. Phylogeny. The phylogenetic tree to the right shows the evolution of OCC-1 among humans and the orthologs from the various taxa that contain OCC-1. The results of this phylogenetic tree follow in accordance with the predicted evolutionary history of animals on Earth. Expression. Expression level. OCC-1 has moderate to high expression throughout the body. Therefore, OCC-1 is ubiquitously expressed in humans, notably high expression in the kidney, skeletal muscle, and pancreas, and low expression in the heart. OCC-1 has shown to be overexpressed in various colon carcinomas. In regards to homologous expression, in situ hybridization data revealed that OCC-1 is expressed in the primary visual cortex of the MAC in an activity-dependent manner. Disease state expression. Profiles from NCBI Unigenes show the expression of OCC-1 in adrenal tumors, chondrosarcoma, gastrointestinal tumors, kidney tumors, leukemia, liver tumors, prostate cancer, soft tissue/muscle tissue tumors, and uterine tumors. Regulation of expression. Promoter. The promoter of OCC-1 is GXB4,407,929 and 601 BP in length. The promoter can be found on the plus strand and begins at 105,234,790 BP and ends at 105,235,390 BP. Interacting proteins. OCC-1 is shown to interact with HRG4, elavlone, CREL, and IRS4. Elavlone functions to stabilize mRNA for gene expression. CREL is involved in lymphoid and cell growth/survival, with a specific presence in T-cell malignancies and cancer. HRG4 plays a role in signal transduction and trafficking sensory neurons and is located primarily in the retina, which relates to the discovery of expression in the brain of the MAC through in situ hybridization data. IRS4 functions as interface between growth factor receptors consisting of tyrosine kinase activity, such as insulin receptors. IRS4 also is involved with the IgF1 or mitogenic signaling pathway. Clinical significance. OCC-1 has shown to be overexpressed in multiple colon carcinomas. References